I do want to talk to improving upstream yields, and I'm curious. Whoever wants to start, Kevin, you're on my screen. What do you think are the most impactful ways to improve upstream yields that you've seen?
Yeah, so my area of focus would be predominantly in synthetically derived peptides, and so there's been a concerted effort, to look at solid phase peptide synthesis and opportunities for improving the quality of the peptides generated in this fashion. And so I think some of the real opportunities are fall around characterization of the process, so understanding kinetics of coupling, kinetics of deprotection, to more efficiently utilize the time associated with manufacturing, but also optimization around reagent use. Amino acids are generally used in a fairly sizable excess in the solid phase in order to drive the purity, and drive complete conversion on the sequestered peptides. So, those efforts to understand and characterize the chemistry itself to more efficiently use the reagents is helping, drive down some of the costs associated with solid phase peptide synthesis. There's significant quantities of solvent used in SPPS to as part of the intermediate wash steps. So there's efforts to reuse those washes in a way such that washes that contain low levels of impurities can use as initial washes in subsequent steps. And so there's ways around more efficiently using the large quantities of solvent as well. So there is a lot of effort in primarily in the characterization space in order to most efficiently use the raw materials associated with peptide synthesis.
Great, thank you. Matteo, Chuck, anything else?
Well, there is one thing that. Oh, sorry, Chuck. The one thing that I totally agree with Kevin. You need to optimize your process in all the aspect, and also you need to develop monitoring to understand if the endpoint of your process in solid phase chemistry, but not only with the coupling, but for example, the washes that he is referring to, because you are washing away something historically, something from the resin, your wash should be appropriate to remove that material, and you don't want to do washes that do not contribute to the quality of the product. So for example, ways to quantify the residual piperidine, we're talking about technically now, but for example, way to quantify the residual piperidine in your reactor after 2 washes, 3 washes, or 4 washes can allow you to very well control your process to the point where you only consume reagents that are really necessary. And unfortunately, in the peptide field, up to maybe 10, 15 years ago, it was a very standard toolbox of number of washing that you will do without knowing exactly why you were doing so many washes. It was a kind of historical approach to making peptide. And as we move now in a more scientific approach to making solid phase peptide, all this in-process monitoring system help you. And the other thing that I think Kevin will agree with me, there is the problem of the resin. Resin are still a little bit of a black box in terms of characterization of the property of the resin. There are slight difference between different batch of resin that can have an impact on the quality of the final API. So one of the things that I think the industry has to improve on is the characterization of the starting resin in terms of not only the particle side, but in terms of the structure inside the resin. These are some of the things that sometimes keep me awake at night thinking if this new batch will have a problem or not. And there are ways to check if your resin is good with practical synthesis, but the analytic around the resin are much more complicated.
I think, Matteo, that's a great point, because I think any sort of a priori characterization characterization that we do on the resins, other than a direct use test, enough to tell us that a resin is going to deliver the quality that we expect. I think we default down to a conservative approach, which is use testing lots of resin prior to committing them to manufacturing. And I would love to see us invest time and effort into better characterization of the chemistry associated with synthesizing the resins themselves, so that we can predict better the behavior and how it will work once we put it into a manufacturing environment.
Yeah. Chuck, anything to add there in terms of what Kevin and Matteo said?
I won't comment on the upstream portion. I do think it's interesting to compare the approaches of biological processes versus synthetic ones on the downstream side. And just in general, as Matteo mentioned, the peptide world has been the same pretty much forever. I mean, there's been some minor changes that get adopted in terms of, Bach for Zefmoc or whatever, in terms of the synthetic approaches, liquid phase, solid phase. But in general, most people do things the same. And the peptides end up the same, acetate salts. I mean, there's reasons why they're, out of all the commercial products, 99% of them are acetate salts because they're easy, they work. We always we go from TFA to acetic acid or whatever. And being relatively new to the space, I was just kind of fascinated by the fact that it's very homogeneous, which is a good thing, of course, if you just want to come in and knock something out. On the downstream side, it's been interesting to see people doing lots of large-scale reverse phase, which is expensive and not particularly intuitive as to that being the only tool in the toolbox. In biologics, it's not uncommon to use a variety of different chemistries. As a matter of fact, it's almost 100% the case where you use orthogonal approaches, the downstream process to purify your product away from the impurities. But that's not so common in peptides, to use ion exchange, to use other forms of downstream processing. So I think it'll be, and so maybe having some fresh eyes, having some different experiences, maybe some ways to improve yields. Because in general, the separations are primarily driven on efficiency, right? So small particles, large, tall columns, and those are not generally efficient ways. You want to do separations based on chemistry, and I don't see that. So who knows? Maybe I can add some value in the peptide space in the long term. We'll see.
Yeah, that's great. And
Todd,
any other points?
Yeah, I just wanted to add one more comment back on improving the efficiency of upstream back in the solid phase. And I think we agreed there that better characterization tools for more efficient utilization of reagents is key. One of the other items that we've incorporated pretty substantially into our solid phase build is the incorporation of small chains rather than single amino acids to get some improved efficiency. It's not uncommon to use, for example, diproline to jump past a proline pair and avoid the diketopiperazine mechanism of elimination and formation of deletion impurities. And that includes, the incorporation of tetramers and trimers of amino acids that can be synthesized chemically as part of the solid phase build to get a little more efficient utilization. There was some good papers that came out a couple of years ago, Dr. Reddy Lab, et cetera, that published on this. And it's a really great way to help improve the overall efficiency of solid phase peptide synthesis using groups of amino acids rather than single amino acids.
Kevin, we had an audience question coming here from James, who asked, can you quantify how much of an improvement the hybrid phase is over conventional synthetic processes? How much improvement in terms of cost?
Yeah, it's difficult to put that. That's a really, really good question, and it's starting to allude to the utilization of hybrid synthesis technologies where we We put together fragments of the overall peptide in solid phase and then assemble those in liquid phase through traditional solution-based amide bond formation chemistry. I think you can see significant yields. It is very much sequence and asset specific. I've seen linear sequences built on solid phase that gave, effectively a 0% yield at the end of the build because of the those mass transfer resistances and the lower and lower yields over the course of a build. Where in that situation, the hybrid approaches, it was exemplified by Bray in that 2003 article on enfuritide, where you can see substantial improvements in the purity and the yield using hybrid. Put an exact number on it, it's difficult. It's very much specific yield from the same quantities of raw materials and amino acids going to a hybrid route. So I think there's opportunities for significant reduction in the overall cost of synthesis. Keep in mind, it is at the expense of 4 solid phase builds or 3 solid phase builds, depending on the number of fragments one utilizes. But then there's opportunities to maybe get away from those solid phase builds as well. But it's substantial, the improvement I think one can see through a hybrid route.